Alrighty, welcome to part 2 of our two-part lecture on Chapter 14, DNA Structure and Function. So we heard about Chargaff in part 1. Erwin Chargaff was an American biochemist who actually determined the composition of DNA, and what he found was that the amount of adenine, okay, so remember the four bases of DNA: adenine, guanine, thymine, and cytosine, right? A, T, G, and C. And what Chargaff deduced was that the amount of adenine equals the amount of thymine. So A equals T, and the amount of guanine likewise equals the amount of cytosine. So G equals C there. So the ratios, and that the ratios of A to T and C to G vary between species. However, there is always an equal proportion of purines, remember that's the double chain, the double ring bases, and pyrimidines, that's your single ring bases. So DNA, if we look at its structure here, we know that DNA is a nucleic acid. We learned that in Chapter 3, I think, when we talked about macromolecules. And as a nucleic acid, it is composed of nucleotides. So what is a nucleotide? That's a 5-carbon sugar in the case of DNA, 5-carbon sugar called deoxyribose. So that's ribose as in RNA that has been deoxygenated, deoxyribose. There's a phosphate group in there, PO4, and both of those are attached, or rather the phosphate group is attached to the 5' carbon of the pentose sugar, and then there's a nitrogenous base, either A, T, C, or G, and you can see that over here. Now, as I mentioned in the first part, we have these phosphodiester bonds that are linking the sugars of adjacent bases via that phosphate group. And so we end up with a phosphate alternating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. We call that the backbone, the phosphate sugar backbone. That's all covalently bound. It's very strong, and that's what keeps DNA structurally sound. And then connected to that 1' carbon of the sugars are those nitrogenous bases. You see them there. Okay, and this is one side. Remember, DNA is double-stranded, right? So this is one side, and then the other side, we call it its complement, would be built the same way, but wherever we have an A, we'd have a T, or an A, we'd have a T, and a C, we'd have a G. That's how that works. And so here we see it. So purine always bonds with a pyrimidine. So you never have a purine and a purine. That's a double ring and a double ring connected. It's always a double ring with a single ring. And so that limits the opportunities right there. And so here we see G always binds with C, and A, adenine, always binds with thymine, and they're connected together. So remember, these guys are connected to the phosphate sugar phosphate backbone on either side. But these bases are connected together with hydrogen bonds. Now, one hydrogen bond, we learned that early on, is a very, very weak bond. However, we're talking about thousands and thousands and thousands, tens of thousands, hundreds of thousands of hydrogen bonds holding a single length of DNA together. So that's a lot of power there. However, individually, there's no strength in a hydrogen bond, or very little. But when you talk about that many hydrogen bonds, there's a lot of strength there. Okay, so purine always binds with pyrimidine, and we call that complementary base pairing, where A always binds to T, C always binds to G. Adenine forms 2 hydrogen bonds with thymine, and cytosine forms 1, 2, 3 hydrogen bonds with guanine. And that allows for consistent diameter of 2 nanometers, and I'm not concerned that you remember that. So, but remember that DNA is a double helix, and so here we see the sugar phosphate backbone on this side and the sugar phosphate backbone on that side. And remember, in the first part we learned that there was a polarity to a strand of DNA, 5' to 3', and that's going to be important when we talk about replication, but it runs in the 5' to 3' direction. And so the opposite strand is going to be runs in the opposite direction. So if this strand is 5' to 3', then this strand is going to be 3' to 5', or 5' to 3' going in the opposite direction, right? It's like a two-lane highway where one lane is going north and the other lane is going south, right? But they're parallel. We call that anti-parallel or anti-parallel, where one is going in one direction and the other is going in the opposite direction. Okay, strands are anti-parallel. One is 5' to 3', the other is 3' to 5'. Phosphodiester bonds within the nucleotides. All right, so or I should say between the nucleotides, phosphodiester bonds hold these guys together, the backbone together, and hydrogen bonds hold the bases together. These are covalent bonds in here, by the way. Let's go to the video from Fun Biology. This is DNA replication. Since the remainder of this Part 2 lesson is going to be on DNA replication, here's a video that illustrates for you, animates for you the process of DNA replication. And then I love Professor Dave Explains videos. So here is Professor Dave explaining DNA replication, copying the molecule of life. So that it just gives you two different perspectives on the same subject, DNA replication. All of these links are in your embedded videos page. Okay, so now that you've watched the videos and you understand what replication is all about, let's just review. So replication is a duplication of DNA. Remember, as I told you in the first part, this occurs in the S phase, the synthesis phase of interphase of the cell cycle. Now, we say that replication is semi-conservative because remember that DNA is double-stranded. And in order to get to the bases, which are tucked in between the sugar phosphate backbones, right, we have to sort of unzip it. We have to remove those hydrogen bonds or break those hydrogen bonds and unzip the two strands. And then once the two strands are separated, then we can, not we, but we can have, the cell can send enzymes in there to then replicate each strand. We call it semi-conservative because you have two strands. Once you zip them apart, you have two strands, and each strand serves as a template for a new strand. And so you get two new copies of the DNA, the parental DNA, if you will. Each has one original strand from the parent, and each has one new strand that we've just replicated. So we call that semi-conservative because one strand of the parental DNA is conserved. Each daughter cell gets one parental strand, that's the conserved strand, and one new strand. Now replication requires the template, or the template, which is going to be the two strands of the original parental DNA. A primer, that's going to be RNase. It's an enzyme, RNase, right? It's an enzyme that puts an RNA primer onto the template strand. We need that so that DNA polymerase, right, which is an enzyme, ASE, can come in there and lay down new nucleotides. And so we see the sugar phosphate backbone of one side here, and DNA is coming in. We don't see DNA here, but DNA polymerase happens to be polymerase 3. There's a polymerase 1, a polymerase 2, and a polymerase 3, and I'm not concerned that you can differentiate between the three, but just if you're reading your text, you run across polymerase 3, and you're thinking, well, what the heck is the difference? Well, polymerase 3 is the one that lays down new nucleotides onto the growing new strand, right? And so here's the nucleotide. Here's a T that's going to connect to this A, and it's DNA polymerase 3 that brings this nucleotide in, removes a couple of the phosphates, because remember, the phosphate group is a P and 4 oxygens. So it's going to remove, and it's going to take this phosphate group, and it's going to connect it together to the free OH group on the adjoining or adjacent sugar. All right, and that's the pyrophosphate bond that gets formed. DNA polymerizes that pyrophosphate bond, and once the nucleotide is in place and connected, then the little hydrogen bonds can form. You see them there. Two little hydrogen bonds can form between the T and the A. We're going to look at this in several different ways and explain it in several different ways, and you're going to get so sick of replication or hearing replication that you're going to know instinctively the next time you hear replication what exactly is happening. So we have three stages of replication: the initiation stage, the elongation stage, and the termination stage. And that's just like anything. All of these molecular processes that we've been talking about all have an initiation stage, an elongation stage, and a termination stage. So that's nothing new. So in order to initiate replication, remember, we have a double-stranded DNA and it's all connected and wound up. So the first thing we need to do, if you think about it logically, and the bases are tucked inside between the sugar phosphate backbones, and that whole thing is twisted together like a spiral staircase. In order to get to the stairs, we have to untwist the staircase or untwist the ladder, and then we have to open it up. So that's what's happening in initiation. So we have an enzyme that comes in here, and we say it unzips the DNA, the double-stranded DNA, unzips it, and so here we end up, there's one strand there, and there's another strand there. They were initially twisted to get connected and twisted together. So we've broken the hydrogen bonds, or we've untwisted the ladder, right? And we've broken the hydrogen bonds between the rungs of the ladder. We call that unzipping. Notice here the arrow. Unzipping occurs in this direction. And so now we have a template strand over here and a template strand over here. Remember, in order to replicate, we need template, we need primase, and we need polymerase. Okay, and so then elongation is when DNA polymerase comes in and adds on the corresponding nucleotides to grow the new chain, the new side of the semi-conservative DNA chain. And then termination, we cut it off and we sew everything back together and we twist it all back together and we move on. Now you'll notice that in this particular illustration, we'll get it closer in a minute, regarding elongation. So initiation is we're going to untwist the ladder, we're going to unzip it. That means we're going to break those hydrogen bonds. We're going to unzip it. We have a special enzyme that does all that. And then DNA polymerase comes in here. Well, first of all, I should say the RNA primase comes in here and puts down a little RNA primer, because polymerase cannot jump in and just start laying down nucleotides corresponding to the template strand of DNA. It needs an RNA primer there to begin with, and then it can start off of that RNA primer, that's the primase, then it can start laying down corresponding DNA nucleotides. Okay, so that's how that works. And so what happens here, remember, replication is always in the 5' to 3' direction. We call this the leading strand because the replication here is continuous. In other words, we'll get to these breaks in a minute, but you see how you see these little breaks down here? You see these little segments down here? That doesn't occur in this continuous strand. We call this the leading strand because it is continuous. There are no breaks in it. And you'll notice that replication is in the 5' to 3' direction towards the replication. We call this the replication fork. Once we untwist and unzip, then we have this fork there, like a fork tongue. Like a snake tongue, we got this fork here, and the leading strand is always replicated towards the replication fork. We call it the replication fork. Now, we call this the lagging strand because it involves discontinuous replication, which means that, remember now, replication is always in the 5' to 3' direction. So that's why we have to go in reverse, as it were, in the lagging strand. Because remember, the lagging strand, you know, this is the 5' end. So if this were a continuous strand, then we would be replicating 3' to 5'. Okay? So in order to maintain the rule, we have to replicate 5' to 3'. So as a result, the lagging strand, and it's called that because it lags just behind the leading strand, it is replicated in a discontinuous fashion in these little segments. And then we got to come back and tie these segments together. Let's look at it a little bit closer. Okay, we'll get to it in a minute, but hold that thought. All right, so here are your players in initiation. Helicase. Now think about it. Helicase, right? Helicase is it's an enzyme that's going to come in here and it's going to unwind the DNA. So remember, the DNA doesn't look like this, right? It's twisted together like a spiral staircase. So we have to untwist it, and then we have to unzip it, and that's the job of this enzyme helicase. You should remember that. All right, so we untwist it and then we unzip it. That's the job of DNA helicase. Now that we have these free ends, if we didn't do anything to hold them apart, these hydrogen bonds would just, these bases would just automatically anneal. We say anneal back together. So they would bind back together. Those hydrogen bonds would just automatically reform. So we have to hold them apart until we can do the replicating. And so we have other proteins that come in here. We call these single-stranded binding proteins, SSBPs. That bind to each of the sides of the unzipped DNA, and that keeps them apart. It's sort of like a little anchor that keeps these strands apart, keeps them from re-annealing. And then we have an enzyme called gyrase, which prevents supercoiling. So an enzyme that comes here. This says topoisomerase is sort of a generic term. Gyrase, it keeps the DNA, because remember, we're unzipping the two strands in this direction. So as these two strands are pulled apart, I don't know if you ever played with rope or string. You know, you cut a rope And rope is made up of, or string made up of 3 or 4 smaller strands. And if you start untwisting them and then pulling them apart, as you pull apart and as you open that's a fork, as you open that fork up, the rest of the rope starts to bunch up, right? Well, that's going to happen with DNA as we pull these apart in this direction. As we unwind them in this direction, it's just like the string or the rope. These strands back here are going to start bunching up and they're going to get all tangled. And we call that supercoiling. So gyrase here latches onto the double strand and prevents supercoiling of this stuff back here. Because if we supercoil it, then we can never get it unwound and we can never get it replicated, and that's a big problem, obviously. Okay? You should remember those 3 players. So elongation occurs, first of all, when RNA primase, here's the RNA primase that makes a short little primer, 5 to 10 bases long, on the template strand of DNA, because DNA primer cannot just jump in here and start laying down nucleotides. So we have a little primer that comes in here, lays down a little RNA primer. That's the red oval here, red football. And then DNA polymerase 3 can come in here. See, there's the primer in yellow. Then DNA polymerase 3 can come in here and start laying down. It uses the RNA primer as like a starting point, and then it begins laying down the appropriate complementary DNA bases. Later on, we have an enzyme that comes in here, clips out the primase, and lays down those DNA nucleotides. But the point here is DNA polymerase 3, by the way, matches existing DNA bases with complementary nucleotides. Remember, A to T, C to G, and links them together. And there are several common features here. The new bases are added to the 3' end of the existing strand, right? The 3' end, because remember, we are replicating 5' to 3', right? 5' to 3'. So we're going to add to the 3' end of the growing chain. A replication again, synthesis occurs 5' to 3'. And each requires an RNA primer. That's the pink football here. Now, DNA polymerase can only polymerase 3 can only synthesize in the 5' to 3' direction. And so the leading strand is synthesized, as we've already learned, continuously from the initial primer. The lagging strand is synthesized discontinuously with multiple primate events. So I'll show you the picture again in a minute, but remember that one illustration. There are segments in the lagging strand. There are segments, right, of DNA, and they're separated. So we call that discontinuous replication, and it happens a little bit behind, time-wise, a little bit behind the leading strand. That's why we call it the lagging strand. And those segments, eventually we have to go back in with another enzyme called ligase to ligate them, to stitch them together to make one whole length of DNA. And those fragments are called Okazaki fragments, named after, of course, Okazaki, who originally described them. Now, it says the lagging strand requires multiple primate events, because for each of those Okazaki fragments, needs an RNA primer. So primase has to go in, lay down the primer, polymerase comes in, lays down the bases. Then RNA lays down a primer for the next fragment, and DNA comes in and lays down the bases. Then RNA comes in, lays down the primer, DNA comes in, lays down the bases. See how it lags, see how it's sort of segmental. Okay, so the replication fork, okay, I've already described this. The replication fork is after helicase comes in and unwinds and unzips the two parental strands, and that creates this fork. We call it the replication fork. DNA primase. Now here's the deal. DNA primase is an RNA polymerase, okay? I mistakenly was saying RNA primase back there, but it's an RNA enzyme. It's going to lay down a primer, which is an RNA primer, but the name of the enzyme is DNA primase because it primes DNA for replication. Don't let that confuse you. Just remember it's a primase, but it happens to be an RNA polymerase. But it's priming DNA, so it's DNA primase. Okay. And then, so later, RNA will be removed. Those little segments of RNA primer will be removed and replaced with DNA by DNA polymerase 1. That's the job of polymerase 1. All right, so here's a larger illustration. So there's your fork. Here is your one parental strand. Here's the other parental strand. The leading strand here is replicated in the 5' to 3' direction towards the fork. And so DNA primase lays down an RNA primer here first, and then DNA polymerase 3 can come in and start replicating that strand. It's continuous, it's non-stop towards the fork, and that's why that's called the leading strand. Okay, so replication occurs in the 5' to 3' direction. So here we have to start at the fork. This is replicating towards the fork. With the lagging strand, we start at the fork. And by comparison, you will think we're going in the opposite direction, okay? And that's why it's lagging. We're going in the opposite direction. So we have to make a segment, make a segment, make a segment, because the whole time we're making this segment, the fork is opening up, right? Helicase continues to open up the fork in this direction. And so we're going to make a segment, and then we're going to jump back here and make a segment, and we're going to jump back here and make a segment, and jump back here and make a segment. So always going back and moving 5' to 3' for 3', go back 5' to 3', go back 5' to 3'. And so really what you see here is, okay, this is stop motion, right? So this is just a picture in time. But really what is happening here, RNA primer, DNA primase lays down an RNA primer here. Polymerase comes in, replicates 5' to 3', this segment, this Okazaki fragment right there. Okay, right there. So, but while that is happening, the fork is open more. Helicase is doing its job, and it's opening the fork up more. So now that primase has to come back and lay down another primer. Polymerase comes back and lays down complementary bases. Okay, the fork is continuing this way. Primase comes back, lays down a primer, or primase comes in, lays down a primer. Polymerase comes back and lays down bases. All right. So really what is happening here, we see this, we see 3 segments for illustration purposes. All right. But really what is happening is primase comes in, lays down a primer. Polymerase comes in, lays down the bases in this direction, 5' to 3'. Primase comes in, lays down a primer. Polymerase comes back and lays down bases. Primase lays down a primer. Polymerase comes back and lays down bases. So it's always sort of this like returning polymerase cycle where polymerase always has to jump back and lay down bases and then jump back and lay down bases because, you know, the leading strand and the helicase is always working in this direction. So DNA polymerase 3 on the lagging strand always has to jump back to catch up, and then jump back to catch up, and then jump back to catch up. And as a result, you end up with these short segments, these Okazaki fragments, short segments of replicated DNA, but you'll notice that they're separated. So now we have to have an enzyme that will come in here and stitch these back together so we have one continuous strand. And that enzyme is called ligase. We call the process ligating. It's like when you go to the you cut yourself and you go to the hospital and you get your the skin stitched back together, sutured back together. They are ligating, right? They're tying these back together so we have one continuous strand. And also it will remove the RNA primers, because remember, each one of these has an RNA primer or a DNA primer connected to it. RNA primer. DNA primase does the job. Okay, RNA primer. All right, so those have to be removed, and then these fragments have to be stitched back together. In the meantime, replication continues on the leading strand, 5' to 3', never stops. So we call it the leading strand. It's continuous. So discontinuous synthesis by DNA polymerase 3 away from the replication fork. Remember, the leading strand is 5' to 3' towards the fork. The lagging strand is 5' to 3' away from the fork, and so polymerase 3 always has to jump back. All right, always has to go back and lay down bases. So each lagging strand, each Okazaki fragment requires an RNA primer, and all RNA primers must be removed by DNA polymerase 1. So DNA polymerase 1 will come in, remove the RNA primer, lay down that 5 or 10 missing DNA bases, and then DNA ligase will come in and stitch them all together. Okay, this is just another look at the lagging strand being replicated. So the DNA polymerase 3, that's DNAP3, synthesizes DNA. DNA polymerase 1 replaces the RNA primer with DNA. And DNA ligase joins the Okazaki fragments together. And that's all this illustration is showing you. Okay. Anything new here to look at? The leading strand requires one RNA primer, and replication is 5' to 3' towards the replication fork. Each Okazaki fragment on the lagging strand requires an RNA primer. Polymerase, so that's DNA primase. Polymerase 3 comes in, lays down the DNA bases in the 5' to 3' direction. And polymerase 1 will come in, remove the RNA primer, lay down those bases, and ligase will then come in and tie these Okazaki fragments together. This is interesting in that don't think this just happens just at one point until the whole thing is replicated, right? We have in this case, you see this bubble here. And so we have a fork going in this direction, a fork going in that direction. We only looked at one side of it to describe the process here, but you can see that the replication begins at the origin of replication, proceeds in both directions. So we have an origin of replication, and the replication can go in both directions. where on one side you have a leading strand and a lagging strand, and the other side you have a leading strand and a lagging strand. And we call that the replication bubble. So we actually have this bubble. We have one point, one origin of replication, but then as the two strands are pulled apart, then we get a fork on either side of the origin of replication, and so that replication can occur in both directions. So here we see this really cool micrograph where you see DNA replication occurring. Here's your DNA, right? There's the double-stranded DNA right there, double-stranded over here. The origin of replication is roughly there. It gets pulled, the two strands get pulled apart, and so you end up with a fork here and a fork over there, and DNA replication is occurring in both directions. That's your replication bubble. Okay, so real quick, eukaryotic replication summary. It's more complicated than that same process in prokaryotes, as we'll see. Multiple linear chromosomes, so this is occurring many times, right? Because you have 23 chromosomes to take care of during the synthesis phase of the interphase of the cell cycle. Requires multiple priming events, one for each leading strand, but many for each lagging strand. And so there are multiple origins of replication because these things are really long, right? So here you see one chromosome, and there's an origin of replication going in two directions. There's an origin of replication going in two directions, and same here. And so eventually, right, this fork will meet that fork, and the appropriate enzymes will come in and we'll tie those two together. Versus prokaryotic replication. So remember, prokaryotes, like bacteria, like E. coli, have a single circular chromosome. Here we see it, right? And there's one origin of replication, but you can see that it occurs, replication proceeds in two directions, so that eventually, that's what these little balls here represent. Replication, eventually they're going to come around and they're going to meet over here, and we call that the point of termination. So here's the point of origin, the point of termination. And eventually these replication forks are going to meet over here at the point of termination, where then the two strands here of the new chromosome, which is still semi-conservative, but the new chromosome, where the two ends of the new chromosome are ligated together and then separated, and then you end up with two circular chromosomes, one going to each daughter cell. So we have a single origin of replication. Replication is bidirectional, and termination occurs opposite the point of the origin of replication. Okay. If it's broken, then, well, fix it. So errors do occur during the normal course of replication, and DNA polymerase has proofreading capability, right? So as it lays down a base, and then it double checks that base and makes sure that base is the right base to put down. So it's going to lay, if there's a T, it's going to lay down an A. If it lays down a G, then it's going to halt, and it's going to say, well, that's not right. It's going to pull out the G and put in an A. So it has proofreading capability like that. DNA can still be damaged, radiation and chemicals. We'll look at some of those mutagens in a minute. And mutagens are anything that can increase the number of mutations above acceptable background levels, because mutations are always occurring, and that's the source of evolution. Okay, so Professor Dave explains mechanisms of DNA damage and repair. Good video for you to watch, a little bit long, but good for you to watch to get a really good overview of what's going on as far as how DNA can get broken, and then how the cell repairs that DNA. Types of mutagens include radiation, like X-rays, UV radiation from the sun, of course, chemicals from cigarette smoke, and preservatives in food, and then obviously from pathogenic agents can cause DNA mutation. So DNA repair, roughly two categories: specific repair, non-specific repair. Specific repair, where a single kind of lesion it gets repaired and only that lesion gets repaired. So a specific type of lesion is during proofreading, when DNA detects a base that's the wrong base, then it will stop and it will pull out that base and it will put in the new base. That's a specific repair, versus non-specific repair, where a single mechanism repairs multiple kinds of lesions. So you have some DNA polymerase III, as it is replicating, it can take care of specific repairs, but then there are some mechanisms that take care of non-specific repairs. It's just, here's a problem, take care of the problem. So photo repair is a specific kind of DNA repair. It repairs thymine dimers, sometimes you call these thymer dimers, and they result as and they are caused by exposure to ultraviolet radiation. So you go and you hang out in the sun too long and you get thymer dimers, thymine dimers, and that's where two thymine residues in adjacent nucleotides get bound together. All right, so we know they're already, T's are already bound to the A's, but because of ultraviolet radiation, the photons come in here and they bounce around and they cause those thymine residues to then stick together. It's called a thymine dimer. And photolyase absorbs visible light, uses energy to cleave the dimer. So this thymine dimer is a problem caused by exposure to ultraviolet radiation, and photolyase will then come in, cut the thymines apart, reattach them, and make the repair. There's the photolyase, the big enzyme, and there it is doing its job, and there's the repaired DNA. Excision repair is non-specific. The damaged area is excised or cut out and repaired on the spot, and we have 3 steps involved there: recognition of the damage. So this enzyme comes along and it's looking for damage, and if it finds an area of damage, doesn't matter what kind of damage, it just finds an area that's not right, it's going to excise, we're going to cut that area out, and then another enzyme will come in and repair that area. So we're going to recognize the damage, removal of the damaged region by the repair complex, and then resynthesis of the new segment by, guess what, DNA polymerase. This is DNA polymerase II. And it's non-specific because it doesn't matter what the damage is. If it's not right anywhere along the way, so you have this enzyme going along, and anywhere it finds damage in the DNA, it stops, it cuts out the damaged part, and then it moves on. And then polymerase comes in and repairs that hole. Okay, preguntas? Do you have any questions? Contact me, and that is the end of Chapter 14. See you next time.